So just in case, any of you were wondering what I'm up to when I'm not lecturing here, this is at least what one website thinks I'm doing. Killer alien space viruses, wipe out humanity. Was it a plot of a horror film, et cetera, et cetera. But down here, there's this guy, biologist Prof. Stedman from Portland State University. It's like, wait a minute, huh? What the heck? These guys don't have a clue at all what I wrote about. They just made up a bunch of stuff. So very fake news, which is interesting. And I think you just have to be careful about what you see and what you read online, because we all know it's all true, right? Okay, so that being said, there's another piece of important information here which I apologize for. Mea maxima culpa. This has to do with the fact that some of you may have noticed that the timing for this class got switched a little before registration actually happened, and that didn't get changed in the syllabus. So there was some confusion about when literally what times the midterms are going to be happening. Midterms happen here in class, the normal class time. The dates are no different, the 31st of January and the 23rd of February, but it's from 9 to 10 in here. So someone, I think it was Alex, pointed this out, and there was some confusion about that. Yeah, midterms, dates are just everything that's been in the syllabus. The times are here during normal class time. I'm not sure whether we'd ever have another time to be able to work this all out. So questions about that, worries, etc. There was some discussion about DRC and accommodations as far as that was concerned. So I just wanted to make this hopefully perfectly clear. Maybe that's scarier than the alien killer viruses, but I'm not sure. So today we're going to continue to talk about DNA structure, but really in particular talking about chromatin structure, because DNA as we have it in Watson and Crick's model is not the way most DNA is most of the time, particularly if we're talking about inside of cells and when that DNA is actually going to be being used to get that genetic information and have it be available. So we'll start out talking about nucleosome assembly, which is putting together the individual nucleosomes to make the beads on the string, which you can see down here. That's what we know the most about. One of the things that's really important about understanding how these fit together is it turns out that a lot of the modifications that happen, which are really important for turning genes on and turning genes off, have to do with modifications to these histones. And this gets back to what we talked about last time, which is chromatin, you've got to compact the DNA, but at the same time have it be accessible for all of the other things that are going to be interacting with it, for the most part, different proteins. So this modification is really very, very important. Another thing we'll talk about today is heterochromatin formation. We've got euchromatin or true chromatin, which is sort of open chromatin, and heterochromatin, which is really compacted. And this really has to do with exactly the same thing. Now, compaction, but at the same time also allowing accessibility. Heterochromatin is extra compacted, not accessible, not going to be active. Euchromatin, accessible and active. We'll talk a little bit about that. And then last time, I think I mentioned, I didn't go back and listen to the tape, but pretty sure that I mentioned that we knew very little about how metaphase chromosomes are put together. And then I went back to my office and opened my email and saw that we actually know a lot more about how metaphase chromosomes are being compacted. So this is classic with this course, and also why I mentioned right at the beginning, the textbook is completely out of date because stuff that happened yesterday is stuff that we should have been talking about here. So if we have a chance, that'll be towards the end of today or on Wednesday. We'll look at some really amazing videos on how, or I should say an animation really, of how we think this is actually happening now. I want to start out a little bit talking about the nucleosomes and the process whereby they get formed. But to do that, I think it's important to talk about how people figured out how nucleosomes really were. Now, zome, real quick aside, is something we're going to be talking about a whole bunch throughout the rest of the course. Zome is just going to be a combination of nucleic acids and proteins. So the nucleosome, the ribosome, the spliceosome, et cetera, et cetera, et cetera. So the nucleosome is nucleic acid plus protein. And it's the very first level of compaction that happens in any kind of eukaryotic cell, and it turns out some archaea as well. How do we know about it? Basically, you take purified DNA, which is shown up here at the top, and with that DNA, you treat it with a small amount of nuclease, known as micrococcal nuclease, so that's described here, and then look at how big the pieces are of DNA that you get out after you've done this small amount of digestion. Now, the micrococcal nuclease, just like the vast majority of enzymes, is a protein, so it's relatively large relative to the DNA, so it's only going to be able to cut the DNA where it's not being protected. And so the protection here turns out to be because of the nucleosome and the proteins that are involved in that. So what do you see if you do a treatment with a very small amount of micrococcal nuclease and then just look at the sizes of the pieces of DNA? We'll talk more about these kinds of gels later on, but basically it's just separating your DNA based on size. This is a known size DNA, so what happens to the size of DNA? You've got a lot at about 150 nucleotides, some at 300, some at 450, etc. And that's because here you've cut just completely around one nucleosome, but since you use such a small amount of the micrococcal nuclease, sometimes you'll cut with 2, sometimes you'll cut with 3, etc. So this is the one nucleosomal DNA fragment, 2, 3, 4, etc. So this is what the DNA looks like. Again, it's about 150 nucleotides. If you cut it very briefly with micrococcal nuclease, then you can also look and see what you have in terms of proteins, and that's the histone core. There are 8 individual proteins that are present in here. They're split up into, incredibly creatively named, H2A, H2B, H3, and H4, which are just those different histones. Since those original experiments where people took the micrococcal nuclease and chopped that DNA into smaller pieces, one of the really amazing breakthroughs in terms of structural biology, now this is going from X-ray crystallography instead of the fibers we talked about last time, which are what Rosalind Franklin was using. Now if you can crystallize whatever you're looking at and then get a diffraction pattern from that, you can then get a much higher resolution structure. And one of the highest resolution structures for a while, for the largest, macromolecular complexes, was the nucleosome. And so this is what the structure of the nucleosome looks like. Again, we've got H2A and H2B, 2 of those, 2 H3s and 2 H4s, with DNA wrapped around them on the outside. So the proteins are mostly on the inside, the nucleic acid is on the outside. If you look at the actual structures of these proteins, they're mostly alpha helices. In fact, 3 alpha helices, 1 long alpha helix and 2 short alpha helices. Turns out that all 4, H2A, H2B, H3, and H4, all have this central structure, which is also called the histone fold. And basically what it is a dimerization motif where you get these 2 proteins, in this case, red is H2A, orange is H2B, H2 and H2B come together, H3 and H4 are green and blue. This color scheme we're going to keep using for most of the rest of today's lecture. Yeah?
So will they always be alpha helices?
Okay, so the question, if I understand it correctly, and correct me if I'm not getting you understanding correctly here, is do they always have these alpha helical structures, or do they sometimes change? When they're in nucleosomes, they always have this central alpha helical structure. One of the things that you'll notice is a great lead-in, thank you, is that there are also these extra bits that are not in this central alpha helical region. So the central alpha helical region is almost always, and we'll talk about an exception, of course, a little bit later on, but in general, central part of the nucleosome, the core part of the nucleosome, is going to be alpha helices of each of these 4 coming together, and there are 2 of each of those 4, so you end up with 8. But that's always going to be alpha helical in the middle, and all of them interacting with each other. These tails on the outside of this histone fold, this is where, as far as we're concerned, the interesting stuff is going on. This is where you have lots of modifications that take place, and this is really important, not so much for the compaction. The compaction is happening mostly because of this core structure. It's the modifications that happen out there that modify these core structures in some different way. And we'll talk about that a little bit more later on. One of the things to mention, this is actually not in your textbook, but still same color scheme, is that nucleosomes are very symmetrical. And this is probably really important for packing nucleosomes together. Nucleosomes are great because they can compact the DNA a little bit, but it's only a little bit. And we have to compact it a huge amount in terms of being able to get even interphase chromosomes, let alone metaphase chromosomes. So the only point here is that these guys can really stack on top of each other. Okay, let's look at the, I just lost my connection here again. How these get put together, and we'll see this in a video in just a second here. If you look at how these histones are going to associate with the DNA, the first thing that happens is H3 and H4, the blue and the green ones, come together, and then each of those come together as well. So you have a tetramer, 2 H3s and 2 H4s. Those then bind to a piece of DNA. And as we'll see in just a second, it's going to be a rather non-specific interaction that happens with DNA there. Then, once you have that first set of binding, that starts to wrap the DNA around, and then you have H2A, H2B, which associate with that. And this will be important a little bit later on when we talk about chromosome, I should say more importantly, chromatin remodeling complexes. First you have H3 and H4, and then you have H2A and H2B. In order to get this to happen, you have proteins called histone chaperones, and I just love the thought of chaperones, having 2, well, one preteen and one teenage girl now. Chaperones are just making sure that the wrong interactions don't take place. So here the chaperones are making sure that the wrong interactions don't take place, but you have this association appropriately inside the nucleosome. So let's look at some of those interactions. Here's, again, blue and green. This is H3 and H4 interacting with DNA in that first sort of wrapping interaction here. Main thing to notice here, before we talk about the clicker question, which is next, is that these have lots of interactions, literally over 100 hydrogen bonds. What's supposed to be emphasized here are actually the charge side chains. What kind of charge on side chains would you expect to see in histones? Positive charge. Why? Because DNA is negative, exactly. So there are lots of charge interactions. Are those going to be sequence-specific interactions or non-sequence-specific interactions? You seem a lot less confident about that. So where's the charge? It's in the phosphates. The phosphates have anything to do with the nucleotide sequence? No, absolutely none. So these are going to be non-specific interactions. Yes?
Does it have anything to do specific-wise with the major and minor grooves?
So the question is, does it have anything to do with major and minor grooves? It's a little hard to see in this particular image, but if you look here, there's a major groove, there's a minor groove, there's a major groove, there's a minor groove. You'll notice some interactions here in major groove, some interactions in the minor groove. So it's actually some of both. But if you remember from last time, where do we see most of the sequence-specific interactions? Those are going to be in the major groove. So if we're talking about non-specific interactions, you're more likely to interact with the minor groove. Exactly. So a lot of these interactions are with the minor groove as well. The other thing you notice here is that this does not look like Watson and Crick's structure. It's really seriously bent. And that bending actually turns out to be important for some specific kinds of nucleosomes, and that brings to the point that nucleosomes are not completely random in terms of the sequences that they're interacting with. So most of you probably noticed that I skipped ahead one slide too many, so I need a water break, and we'll have a clicker question. So how many protein N-termini are there in one histone core? And will it start? Yes. Ten, 5, okay, what do we think? I've actually had to, oops, where's our show results? Where are they? Where are my results? Try here. Technology, such a wonderful thing. No. Where are my results? Show results. Oh, no base station. Thank you. It's here. Really, trust me. It's a base station. Well, be that as it may. So the numbers that you got are 11%, no, 11 for 1, 3 for number 2, 37 for number 4, 87 for number 8, and 3 for 16. So I spent a long time thinking about the right way to write this question, and it really has to do with the number of individual proteins that are in that core. And you can argue about what different sequence proteins, if you talk about what different sequence of protein, how many different histones you have in there. 4 different histones, but there are 2 of each of them, and each of those is an N-terminus and a C-terminus. So what's the correct answer? If I could actually get this to show itself, it would be 8. And I should be able to see these in later, and I'll check. If it turns out that this didn't work, everybody gets points, because I don't know which 87 you were. Again, we're still having trouble finding the base here. So, well, turn it off, turn it back on. See if that'll work. Okay, now it's finding it. Very strange. Okay. Next slide. So I mentioned the sequence. Oh, sorry, Devin. Yeah. So the question is counting N-termini equivalent to counting the number of tails? That would be one way of talking about it as well. But the idea here was basically number of polypeptides that are present in the nucleosome core. Okay, so again, I mentioned last time we looked at that hydrogen bond interactions and the ionic interactions between the nucleosome, and that was just H3 and H4, but it turns out the same thing is true for H2A, H2B as well, is that most of those interactions are going to be in the minor groove, a few in the major groove, and the DNA is incredibly bent. And if you think about bending DNA, it turns out it's actually easier to bend DNA that's got A's and T's and squish those base pairs together than it is to do G's and C's. So if you happen to have a piece of DNA that has a lot of AT base pairs near this minor groove that's going to be next to the nucleosome, and G's and C's out here, which are not as compressed, you remember how many base pairs are there per one repeat of DNA? This is what Rosalind Franklin basically showed. How many base pairs per single turn of B-form DNA? 10. So if every 10 base pairs, it's a full turn from here to here, are A's and T's, then you'd expect this DNA to be able to wrap around nucleosomes very well and actually have somewhat of a sequence specificity for this. Again, generally nucleosomes aren't sequence specific, but if you're trying to do an experiment, we have nucleosomes at a particular place on your DNA, you're going to want to have DNA that every 10 nucleotides has got A's and T's in it. So this helps considerably in terms of setting up these structures. So that's your sequence-specific nucleosomes. And again, for the most part, these are interactions with the phosphates, the interactions with the riboses, not a very sequence-specific, but more of a structure-specific kind of interaction which is happening. If you've got this nucleosome, 150 approximately nucleotides wrapped around the nucleosome core, how the heck can you get, maybe back up one here, a sequence-specific DNA binding protein that you might happen to need for something to bind to sequences which are right here in the major groove if it's wrapped around a nucleosome? Hard to do that, you would think. But when we look at the video in just a couple of minutes here, we'll notice everything's flopping around a lot. And it turns out that's very true in biology. Anytime we look at any of these static pictures, that's one snapshot of what we're looking at here. So they're very dynamic situations. And one of the things that was shown quite early on in terms of how nucleosomes are interacting with DNA, you can set up an experiment where you take a piece of DNA that every 10 nucleotides has A's and T's, and the nucleosome will sit in one particular place on that piece of DNA. And then you put in a DNA binding protein that should be binding to a sequence that's in that nucleosome that should never be able to be bound because it's wrapped around the nucleosome. Turns out that all nucleosomes inside the cell, because we're at 37-ish degrees Celsius, are flopping around. So the DNA comes off of even these nucleosomes that we always see as beads on a string, nice solid stuff, very frequently. So the numbers here are not important, but some percentage of the time, even if you have this DNA sequence shown here in white that's wrapped around a nucleosome, is otherwise non-accessible, you put in a DNA binding protein that can just bind to that white sequence, it can bind because this piece comes off of the nucleosome on a relatively frequent basis. So, 250 milliseconds, 10 to 50 milliseconds, etc. If this DNA binding protein is around, it can bind. If it doesn't, the nucleosome reforms. And so this allows you to have the protection, which is wrapping around the nucleosome, and the compaction, but still have accessibility to other proteins which are there. Yeah?
Is that due to the type of binding between the DNA and the nucleosome? Or the histone binding?
I'm not sure I completely understand the question. Is the fact that you have this binding and unbinding specific to nucleosomes or the histone interactions?
Bonding that happens between the positively charged histone and the negatively charged DNA.
Okay, so the question has to do with, is it just a charge-charge interaction? Are charge-charge interactions those that are relatively weak and come on and off very rapidly? The answer is no. That's not specific to any one of those particular kinds of interactions. Nucleosomes, and it's actually true for all of these binding proteins, are going to come on and off. It's just going to be at different rates relative to each other. And usually the specific interactions are going to be those that are going to stay on a little bit longer. And we talk about these being stronger interactions, which are there. And so in this case, for instance, where you have this DNA binding protein interacting with this one particular piece, this is likely to have a stronger binding to that piece than the nucleosome would have to that particular piece. So this is going to stick around in this form for a lot longer than this nucleosome is going to be bouncing back and forth here. And we'll talk about sequence-specific DNA binding proteins probably ad nauseum later on in the course, how those are interacting with each other. However, that's not enough. And we know that because there are a large number of so-called chromatin remodeling complexes which are present inside the cell. Now, these are complexes, which means that they've got lots of individual proteins that are associated with them. And probably the most important is that they require ATP hydrolysis to do their jobs. And this particular job is a chromatin remodeling. What do we mean by chromatin remodeling? Basically, what we mean is moving nucleosomes around relative to DNA. And that moving could be literally moving the DNA through the nucleosome, which is supposed to be shown here. It's a little hard to see. The blue lines here are supposed to represent particular sequences in your DNA. Now, if you have the binding of this chromatin remodeling complex, the green C, Pac-Man maybe? Anyone remember Pac-Man way back when? So interacts with the nucleosome-bound DNA, ATP hydrolysis happens, and you see this blue sequence gets a little bit closer. This blue sequence wraps further around, and probably most importantly, this blue sequence here, that was otherwise probably blocked from DNA binding, is now outside of the ribosome, and so now you can have DNA binding that takes place here. That's only one kind of chromatin remodeling. There are multiple different ones that happen. Again, all of these are dependent on ATP hydrolysis, because those interactions, even that flopping, the nucleosome breathing, as I talked about last time, small amount of the time the DNA is not part of the nucleosome. Here you have to have this happen to get more pieces of DNA which are accessible to the rest of the world. And as we talk about transcriptional regulation, and it's also true for replication, you're going to need lots of proteins to associate with the DNA. And you need to get nucleosomes as far away from that as possible in order for these replication and transcription complexes to form. Someone asked, I think you'd asked about the alpha helices in the central structures of DNA. One of the things that we're learning, and this is stuff that's happened again since the publication of the textbook, are that some of these chromatin remodeling complexes, what do? They actually take some of those alpha helices that are present in those core nucleosomes and wiggle them around relative to each other. That's what the ATP hydrolysis is doing. The ATP hydrolysis is pulling apart that structure a little bit. And so that's what's supposed to be outlined here. All of these abbreviations are not important, but here is a particular binding sequence. Here's this actually, unfortunately, different color coding. It's histone H3 and histone H4 here. In the presence of one of these chromatin remodeling complexes called SNF2H, again, the name is not important here. In the presence of ATP, all these things require ATP hydrolysis, this starts to flop those core histone structures around relative to each other, basically moving them around. And that moving them around allows this DNA to move a little bit, and most importantly, this little green piece, that's the green piece of DNA that you want to have another protein that's associated with. So this is we're now learning slowly more about how these chromatin remodeling complexes work. Before it was Pac-Man plus ATP does something. Now we know that it's changing the structure of the core nucleosomes in order to get that to actually happen. This is an overview of some of that. Excuse me, yeah.
So the chromatin remodeling complex is specifically acting on the histones that then kind of shift the chromatin around?
Yeah, so maybe back up a little bit here. But so yeah, thank you. And I'll paraphrase it, and I may or may not get it right, so correct me if I'm wrong here. The idea here is that this, and I should have mentioned this before, is a particular kind of chromatin remodeling complex. There are lots of different ones, which I'll talk about on the next slide. But this particular one changes the structure of the core nucleosome particle in order to allow that DNA to move. Now, it's not specifically, it's not the nucleosome itself that seems to be moving it. It's just allowing it to be able to be moved. And probably what it's doing is, we talked about that nucleosome breathing. We just have pieces of DNA coming off spontaneously. It's increasing that amount of DNA being released. Okay, so that's one, great for a lead in here, because that's one particular kind of chromatin remodeling that you have, which is literally just the DNA moving relative to the nucleosome. Turns out that's not enough in many cases. Often you actually have to get rid of the nucleosome completely from the DNA. That also happens through these chromatin remodeling complexes. You can get ATP hydrolysis and the whole histone comes out. Now you've got DNA with no nucleosomes associated with it. Sometimes you can get a whole brand new octamer of DNA. Hopefully you remember when I was talking about the assembly of these nucleosome particles, what happens first? H3 and H4 associate with the DNA, and H2A and H2B come in later. Well, that's what happens in these kinds of chromatin remodeling complexes. You remove H2A and H2B, but H3 and H4 stay there. And so you've got lots of different ways that these things can happen, i.e. just moving the DNA along relative to the nucleosome. Everything stays associated with nucleosomes. And you can tell this by doing these micrococcal nuclease experiments, chopping them up with small amounts of nucleases and seeing that you've got 140 bases all the time, or completely removed. If you chop up that DNA with a micrococcal nuclease, that DNA would be completely gone. So it really depends on which one of these is going on. And one of the things that I was really kind of surprised at is that, again, we think about nucleosomes as being a pretty static kind of compaction of the DNA. This kind of thing is probably happening every couple of hours for all the nucleosomes. Literally millions of nucleosomes in every one of our cells is popping on and off every couple of hours, which is really kind of amazing. Yeah?
So, can we get to understand? So, there's a DNA lacking nucleosome. I thought the nucleosome was specifically DNA with the histone core. So, like, they're in the middle of the bottom.
Yeah.
So, is that lacking nucleosome? Is that what the histone work? The nucleosome treats the DNA with the histone core, and then everything is just core, or?
Yeah, so this is basically kind of a semantic question. Exactly, yes. Those nasty Stedman clicker questions, or exam questions for that matter. Yeah, no, so you're exactly right. When I talk about nucleosome, I'm going to talk about the DNA plus protein as being the nucleosomes. Here, it's supposed to be more of a general case. This is how I understand it. This is not my figure. It's from the textbook, mea non culpa. But the idea there, I think, is that it's that particular DNA which is lacking in nucleosome relative to all the rest of the DNA, which hadn't had this nucleosome remodeling complex, chromatin remodeling complex associated with it, which still does have those nucleosomes. So it'll be a piece of the DNA that is not part of a nucleosome. And I would say here, DNA that's not part of a nucleosome, which would be a better way of describing it. So write to the textbook, tell them they need to change the figure. Okay, so the nucleosomes, again, we understand quite a lot about these individual nucleosomes. We know to a very high resolution what these structures look like. We know a little bit about how the nucleosomes get moved on and off, DNA moved, etc. But most of the DNA in an interphase nucleus is not this nice beads on a string thing. It's really much thicker than that. And so this is a little bit harder to see here. But if you look at the top image, this is an electron micrograph. Each of the big dots here represents a nucleosome. This thin thing between them is the DNA between each of those nucleosomes. This is your beads on a string, individual nucleosomes. But most DNA, if you look at an interphase nucleus, is much more like this, where you have the nucleosomes really packed on top of each other. It's called the 30 nanometer filament. And this is what the majority of your DNA, and I should say in euchromatin, looks like, active chromatin. How do you make these 30 nanometer filaments? It turns out that we understand a lot less about how this works. One thing that's very important is histone H1. Now, one of the things that I neglected to mention when we talked about the histone folds, you remember H3, H4, H2A, and H2B. It turns out that those sequences, not surprisingly, have lots of positively charged amino acids in them. The other thing which is not surprising is it turns out they're incredibly well conserved. If you look at any eukaryotic organism, be it yeast, be it Giardia, be it humans, the sequences of the histones are almost identical to each other, which is not surprising because we all have DNA, same kind of problem, same kind of compaction that has to take place. On the other hand, histone H1s, which is one of the other histones, not part of the core, these are a lot more variable from organism to organism. And that tells you that there's some different things that are going on in the compaction of DNA beyond the individual nucleosome level. So these histone H1, that's what I mean by many kinds, they're not as well conserved. They then help to compact those DNAs like you have up here down to the DNA that looks like here. There also are many other DNA binding proteins that are part of chromatin. So let's back up a little bit. Remember chromatin I talked about last time? Chromatin is DNA plus proteins. That's histones, which is the vast majority of those proteins, but a lot of other proteins as well. So chromatin is not just nucleosomes, it's all of these other proteins as well coming together. And we'll talk about some more of those, particularly talking about metaphase chromosomes a little bit later on. There are a number of models for how people think this 30 nanometer filament forms. This is just one of those particular models where you have nucleosome up here, nucleosome down here, stacked on top of each other. People think this is what this actually looks like. The answer is there are probably lots of different ways that you can form these 30 nanometer filaments. We do know that if you take the core histones and chop off their tails, you don't form 30 nanometer filaments. So it turns out that those tails are also important for nucleosome-nucleosome interactions. And that you can hopefully see here, again, in a cartoonish form. Here we have the tail of one of the core histones interacting with core histones in the next nucleosome, and so on and so forth. And this is just a reminder, these tails really do stick out of this core nucleosome structure, so lots of things can happen to them. If you take an electron microscope image, you see a lot of these blobs, and people try and interpret these blobs and make them look like this. Again, we don't know in detail how these things are interacting with each other. I did want to take a look at this video. Probably most of you have seen this before. It's a nice review, particularly of how the first interactions happen between the individual core histones and the DNA to form the nucleosome in the first place, and how we think some of these core nucleosomes are associated with each other. See if we can get this to actually work. Oh, that's, yeah. This is the wrong image here. Shift this over. Stop. See, it's me trying to use this. That's my daughter winning a cyclocross race, but that's a different story. We don't have to look too much more at that. Okay, let's do the whole screen here. Quick shout out here, DNALearningCenter.org. Great place to find some of these animations. It's funded through the Cold Spring Harbor Lab. So here we have the double-stranded form of DNA, and also be wobbling around a lot, as I mentioned before. This is much more a dynamic system. So here we have the DNA, perfectly happy DNA, H3, H4 interact with it. Then you have what? H2A, H2B. And then we wrap around to get our two circles of DNA. Now we've got the nucleosome itself, all of those tails hanging out and flopping around on the outside. Those tails interact with each other, and again, this is an artist's impression of how people think this happens. Now you've got lots of these 30 nanometer filaments. Again, this is what most of the euchromatin, the active chromatin, inside the nucleus actually looks like. This then gets compacted and compacted more and more. This would now be starting the process moving towards mitosis. We get the maximum amount of chromosome compaction taking place in order to have this. One of the things to notice when you go through this animation, if you want to, is the numbers for the magnifications are off. So go through that and check, and again, send them an email, tell them how wrong it is. 3000x is just about right. Some of the other ones are actually incorrect. Then you have metaphase. This is how all of these chromosomes are coming together. You can actually see them, again, finally under a light microscope, coming apart, and this is the cell division process as it moves forward. Okay, let's start back up with these guys. Play. moving back and forth on me. Here we go. Okay, so now we've finally taken care of that. Everyone can get their clickers back out, and I can try and get my clicker to work. See if it'll work better this time. Seems to. Okay, so which of the following... Come on, now it's not. No base? What do you mean no base? Unplug and replug here. Try again. Yay, okay. Which of the following requires ATP hydrolysis? Nucleosome formation, nucleosome breathing, chromatin remodeling complex activity, or if I can't come up with anything else, I'll put A and C and A and B. Ten, 5, okay. Now this time it actually did show the results. Very strange. Quick reminder, those of you who are still not seeing clicker points after today, let me know. Send me an email. We're just down to about 7 or 8 of you, I think, that shouldn't have those things connected appropriately. So which of the following? Yes, it's the chromatin remodeling complex activity. Nucleosome formation does not require ATP hydrolysis. Nucleosome breathing does not require ATP hydrolysis. Only chromatin remodeling complexes do. Okay, I wanted to talk a little bit more about histone H1. We know something about the structure of these complexes. Histone H1, probably a little bit hard to see here, is shown here in green. Basically, what histone H1 does in most of these cases, as well as helping form these 30 nanometer filaments, is take the DNA and wrap it even further around the nucleosome. So if you do an experiment where you take the core histones and DNA plus histone H1, do that same kind of micrococcal nuclease activity, you'll see that there's a little bit more of the DNA which is protected from these nucleases, which would otherwise be breaking down the DNA. And it's about 20 more base pairs that's associated. So instead of 150, you're talking about more like about 170 of these nucleotides which are protected. And again, it's a compaction process that's helping at that nucleosome level, but also these H1s are important for forming these 30 nanometer filaments. So what do you have after 30 nanometer filaments? From the video, you saw that these 30 nanometer filaments wrap around each other as well. That gets you to the larger filaments, 300 nanometers plus or minus. That's the 30 nanometer filament wrapped around itself. That gives you about 300 nanometers. And what this does is these form what people call looped domains. And we'll take a look at them a little bit later on when we look at some of the individual examples here, particularly lampbrush chromosomes and polytene chromosomes that you see in some insects. Basically, these loop domains are separate parts of the DNA. And my apologies here, this is now a chromatin domain, not a domain of a protein. So again, 2 different definitions of domains. There's also domains of life. Again, just to make it ridiculously confusing, I will try and be specific on clicker questions and exam questions. So loop domains, these are parts of your chromatin which end up in one particular piece. Now, why do we care that it's one particular piece? It turns out that that piece of DNA is almost always regulated independently from all of the other pieces of DNA. So it's a structural thing holding together the DNA, which again, we still need to compact it. We've still got to get it into smaller spaces, but also isolating it and insulating it from the rest of the DNA which is present inside the nucleus. And we'll see how important that is when we talk about regulation a little bit later on. But these individual loop domains, seem to be very important for segregating out different pieces of chromatin, different pieces of the chromosome relative to each other. There are about 2,000 of these in the human genome. We can see them quite easily. If you just look at what different pieces you can split them up into, and it turns out that those 2,000 pieces are all being regulated differently relative to each other. Yeah? Mm-hmm. Yeah, so the question is, just so we've got it here for posterity's sake, are these loop domains moving around as well? Are they as flexible as like the individual piece of DNA relative to the nucleosome? Is that more or less your question here? What it seems to be is that that's actually not the case. That particular loop domain seems to be pretty specific. And the reason that we know that has to do with should have brought up the picture we had last time. I remember sort of the starburst form where you got some really dark area in your interphase nucleus and a lot of little strings coming off of it. Talk about those strings. Those are where you've got your 30 nanometer filaments, your 10 nanometer filaments. There's still a lot of DNA, which is in the black bit, which is in the middle. And that's your nuclear matrix. This is protein. And then the DNA associating with those, that's going to be the scaffold, which is holding these loops together, but also the attachment to the nuclear matrix. And the nuclear matrix attachment regions, these MARs, these are that part of the DNA which is going to be associating with the protein, giving you organization and also some more compaction inside of the nucleus. So let's look at some of these. Back up a little bit more. Just another overview of what we're talking about already. Chromatin organization, nucleosomes. Hopefully we've kind of done nucleosomes now, at least they're static original case. We'll look at modifications on those. These beads on a string are great, but they're really dynamic. They normally are going to have the same proteins relative to each other. What are those proteins going to be? The bead in your nucleosome is what? H3, H4, H2A, and H2B. Hopefully not a broken record yet. But it turns out that there are some of these nucleosomes that are quite different. And we'll talk about some of those modifications that happen in nucleosomes a little bit later on. Why is this important? Well, it turns out that some of these modified nucleosomes are really important for gene expression. We'll talk about this after the midterm, so you can forget about it for now. Genome packaging. So again, we talked about compaction. Compaction is great and well and good that you need during interphase, just to get everything inside the nucleus. But if you're going to do the segregation of your nucleosomes, which you have in mitosis, those also have to come together. And then you need specific parts of your chromosome, which allows those chromosomes to be separated. It turns out that alternative histones and alternative nucleosomes are really important for that. So let's look at some of these differences that happen here. Histone tails. These histone tails are important for association of the 30 nanometer filament, but also it turns out that these get heavily modified. If you look at the sequences of these histone tails, not surprisingly, they've got a lot of these positively charged amino acids in them. K. K is what? Lysine, exactly. So lots of lysines, but you also have some serines and threonines as well. Serines and threonines are interesting for what reason? They've got OHs on them. What can you do with OHs? Put phosphates on them. So kinases, you can modify them through kinases. These are all now going to be covalent modifications. What happens to lysines? It turns out that lysines, as well as having this charge, also have side chains which are quite reactive. And so those side chains can be modified, and they get heavily modified in histones. The most common modifications are either acetylation or methylation that happen, and particularly true on these lysines. So acetylation here, the pink ball, methylation here with the blue ball. It turns out there are all kinds of different modifications that can happen on these histones. These are reversible modifications, and they're modifications that happen at the protein level. How do you get this to happen? Well, anytime we talk about covalent bonds, what are you going to need? Those are high-energy bonds. You're going to have ATP hydrolysis. You're going to have transfer of these individual small molecules or different pieces to your DNA. Turns out that these modifications happen on all of these individual tails, but we're mostly going to talk about H3. Why H3? Because it's the longest and the one that sticks out the most, but mostly because it's the one that we've mostly studied. What happens when you've done these modifications? Oh, sorry, I should back up here. This is not what one particular side chain, I should say, one particular tail on a histone actually looks like. It doesn't have all of these modifications simultaneously. These is just an overview of all the kinds of modifications that have been shown to happen. Usually it's some subset of these modifications, and as far as I know, none of them ever had all of these modifications together. So you'll have, for instance, we'll talk quite a bit about this particular position here, K9, so not dogs. This is now lysine at position 9. In the N-terminus of histone H3, so H3K9, we'll talk about quite a bit. H3K9, so histone H3, lysine 9, methyl group, or monomethyl, dimethyl, trimethyl. So 1, 2, or 3 methyl groups have been added to that particular lysine. Or acetylation at K9 of H3. So any one of those particular modifications is going to happen with other modifications as well. Why? Well, that's because it's a signaling process, and in order to notice this signaling process, you have to have other proteins that are going to recognize that. And just like all the other proteins, the vast majority of that protein is going to be holding a few amino acids in appropriate places so you can have these interactions that take place. In the case of histone modifications, we have what are called histone code readers. The histone code is that pattern of different modifications that have happened on side chains. Those have to be recognized by other proteins. These are the histone code readers. So here's an example of one of those histone code reader complexes here. Complex tells you multiple different proteins that are involved here. These all have little pieces of them, which are supposed to be these little, I don't know, semicircles or pieces down here at the bottom that are now going to recognize specific and usually different modifications on different histones in different nucleosomes. If all of these things match appropriately to this code reader complex, it will bind to this piece of DNA. Once it's bound to that piece of DNA, now it can interact. You remember our interactomes we talked about before, all these different proteins interacting with each other? This is one of the ways that you can figure out how all of these things are interacting with each other. And once you know that, it'll give you some idea on their functions. Here we have the histone code reader complex that can associate with another protein, which then will be able to either modify some of the histones that are here, or maybe help bring in a polymerase complex, or some other kind of complex that you need to modify the DNA at that particular position. So these code reader complexes will bind to modified histones and then bring other proteins to this particular piece of DNA. Now, these are not sequence-specific interactions. These are now histone modification-specific interactions. So it's getting those histone modifications that are really important. And these are just a couple of examples here. This can be gene expression, gene silencing, other kinds of functions which can happen through the binding of these histone code reader complexes. Reader and remodeling complexes, turns out that a lot of these readers also are connected to